chlorophyll reaction center. So why is the question mark? So that we will be discussing. So now, once we have understood that we have photosystems, that is photosystem one and photosystem two, right? So there is some important reaction that is happening, which we call it as a light reaction. Okay. So now I have one more question for you, which is a very, very important question. Listen to me carefully. Can you tell me where are these photosystems located? Okay. Where is photosystem one and where is photosystem two located? Do you know? Where can I find this photosystem one and photosystem two? Is there an answer? Where can I find this? Yeah, no. So let's see. Okay. If we have an answer, that'll be great. Chalo. So yeah, nothing. So it's okay. It's okay. So while I was talking about like where are these photosystems one and photosystems two located, it's very, very important. And we will learn about it as we move on. Okay. So let us see what is happening with respect to these photosynthetic components. So this reaction we call it as a light reaction, and another time in another term we call it as Z scheme. Okay, we also call it as Z scheme. So all right. So we can split this Z scheme. Okay, we have two types of light reactions happening. Okay, one we call it as non-cyclic photophosphorylation, and other one is called as cyclic photophosphorylation. And non-cyclic photophosphorylation is called as Z scheme. And we have cyclic photophosphorylation. We will talk about it. So nothing to worry. So I can tell you that in light reactions, there are two different types of phosphorylation is happening. What are those? One is called as cyclic photophosphorylation. Other one is called as non-cyclic photophosphorylation. So let us understand non-cyclic photophosphorylation because it's quite easy to grasp it. Okay. So now we know that our light energy or sunlight is used for the process of photosynthesis. So now what happens when the light energy or the light hits the leaf? So we know that our chlorophyll pigments are very well arranged at the almost just below the cell walls, right, at the borders, so that they can harvest maximum amount of light. Now with this arrangement, the light energy when it reaches our chlorophyll, okay, our chlorophyll A or the reaction center. Now what happens is there are atoms, okay, which is within our chlorophyll pigments. So now these atoms have electrons. So these electrons, they get excited. Why do they get excited? We know that the arrangement of electrons is in such a way that they are around the nucleus in orbitals, right? We call it as atomic orbitals. So they are revolving around this, nucleus of the atom around it. They keep revolving around it. So at a fixed energy, whenever there is an external energy that is just added or it is exposed to a higher energy, I can tell. So whenever this atom receives the higher energy, now what happens is the electrons within this nucleus, right? Now they get excited. So when they get excited, they cannot remain within their atomic orbitals. Why? It's because of the law of quantums. So whenever the electrons receive more energy, they get excited and they go and jump the higher orbits. As they jump from the higher orbits, now the electrons move out of their orbits. So these electrons which move out of their orbits are taken advantage of. So how are they taken advantage of? Now these excited electrons, they are transported through a series of electron acceptors. So let me tell you in simple terms. Now what is happening? The light energy, okay, is utilized by these chlorophyll pigments. It's a very natural reaction. What kind of light energy we require? So we know that the sunlight is very, very common, but a very fixed wavelength of light is used here. What kind of a wavelength? We have 680 nanometers of sunlight. So whenever the sun radiation of 600 nanometer of a wavelength incidents our chlorophyll pigments, so here the atoms of the chlorophyll okay, they get excited. So that makes the electrons leave their atomic orbitals and jump. So once the electrons leave their atomic orbitals, now what happens is we use electron acceptors to move these electrons downhill. Okay. So what is downhill? See here, the electrons got excited in the reaction center. Okay. So now they are transported. So once they're transported, see here, the electrons 2e, wait, I'll just erase the two electrons that we have received, okay, is transported through our, what did I tell, electron acceptors. Whatever are our electron acceptors, that is plastic, say we have pheophytin as one of the electron acceptor, and then we have plastoquinone, which is another electron acceptor, okay, and then we have cytochrome B6 complex, which is another electron acceptor. So what they do is, from this excited 680 nanometers of wavelength, they start transporting the electrons downhill. Okay. So what is downhill transport? So from higher energy to lower energy, they are transported. So this is called as downhill transportation. So once they are transported downhill, how are we calling it as downhill? If you understand the process of oxidation and reduction, or we just call it as, okay, oxidation or reduction. Okay. So oxidation or reduction, or we call it as oxido reductase. Okay. So if we look at the oxidoreductase potential, so here it is going downhill, downhill of the oxidation or oxidoreduction potential. So that downhill transportation of the excited electrons happens through series of electron acceptors. Okay. So oxidation and reduction happen simultaneously, and it keeps moving here. Understood. So these excited electrons are not utilized. Instead, they are transported through a series of transporters. So where does this electron acceptors go? Finally, after reaching the cytochrome B6 complex, okay, so these electrons are transported to our second photosystem. So I know that our photosystems are named reverse. Okay. So here it is photosystem 2, and here it is photosystem 1. All right. So sorry, this here it is photosystem 1. So what happened? uh, our photosystem 2 was responsible for receiving the light, and somehow when the light incidents on our photosystem 2, here the electrons got excited. The excited electrons were transported downhill through a series of electron acceptors, correct? And then these electrons are not utilized, they are just transported across the gradient. So once they're transported, where are they transported? They are transported to the next photosystem. And what is our next photosystem? It is PS1. So once it is coming to the downhill, okay, by the time it reaches the PS1, now this is not in an excited state. Again, the light incidence on the PS1, that is photosystem 1, the photosystem 1 electrons now again get excited. Now this time, when the electrons get excited, again a series of acceptors are utilized for the transportation, okay, downhill, okay, for a purpose. So what is happening here? The electrons got excited in the photosystem 1. The excited electrons in the photosystem 1, here again it goes through a series of electron acceptors. So let us see what of them. We have A naught, A1, which are all some of the electron acceptors. Here oxidoreduction process is happening, or redox reactions are happening. So here what happens? For example, uh, phylloquinone, iron sulfate proteins, ferridoxin, flavoproteins are some of the electron acceptors. You need not remember the name, but it's all right. You should just grasp the names. Okay, you could just know that there are some of the electron acceptors. Okay. So now what is happening, students? So as it goes downhill, the oxidoreductase potential, now it finally what happens? These electrons are used for the reduction of a very special component. So what is that? That is NADP plus. Okay. Oh, sorry. Yeah. What happens? The electrons that are excited by the P700 are used for reducing NADP plus to NADPH. So this NADP plus to NADPH happens through NADP reductase enzyme. So this NADP reductase enzyme, okay, which is present in the stroma. I will just make it here. Okay, this NADP reductase enzyme, which is present in the stroma, what it does is it converts the NADP plus to NADPH, okay, through an enzyme called as NADP reductase. So this reaction happens. Understand this point. Now, when we take the light reaction, why the light reaction even happens is a first call. So for the light reaction happens because we know that there are two important things that the plant has to perform. What is that? The first thing that the plant has to do is, the first thing that the plant has to do is create some energy. So that energy is ATP molecules that we know already. So, and the second thing that why it requires the energy is having ATP is not the ultimate goal here. ATP is one part of the story. And the second part of the story is we need to prepare glucose, correct, right? So for the process of preparation of glucose, two important components are necessary. What are those? One is energy in the form of ATP, and the second one is the NADPH. So this NADPH and the ATP are utilized in the biosynthesis phase where the glucose or the what do you tell metabolites are produced in the form of glucose or starch. Okay, so now let me pause here. Did you guys understand? Are you listening? Students, you understand light reactions? Yes, no? Give me 1 second. Okay. If you tell me that you have understood, then it's really, really helpful. Okay. But again, these are very, very important points to remember. So for that, we need to understand the process. Okay. So I will point out some of the points which is very important for memorizing or remembering. So before that, I need to be sure that you are understanding what I'm talking, or if you can hear me, nothing. Okay. So just give me some sign. No? All right. Chalo. So I will continue. Okay. So let's see if you understand even better. All right. So now what did we understand in the light reactions? In the light reactions, we are talking about something called as photophosphorylation. So what type of photophosphorylation we are talking about? We are talking about non-cyclic photophosphorylation. In non-cyclic photophosphorylation, two photosystems are involved. So what are those? One is photosystem 2, other one is photosystem 1. Correct? So what did we learn about these two systems? These two systems are arranged in such a pattern. Okay. So it is called as a Z scheme. Why it is called as a Z scheme? It goes downhill the oxidoreductase potential, okay, or redox potential. It goes downhill the redox potential. That is why we call it as a Z scheme. Okay. So in Z scheme, what is involved? We have two photosystems which are very systematically arranged. Okay. First comes photosystem 2 and the next comes photosystem 1. So this is the first reaction of a photosynthesis. So what is the byproducts of this light reaction? So whenever we ask what are the byproducts of light reaction, you should tell that it is ATP and NADPH. Okay, so ATP and NADPH are very important byproducts of the light reaction. What are the accessory or additional byproduct of light reaction? It is oxygen. Okay, very important is oxygen, and other one is hydrogen. We will talk about hydrogen, ATP and NADPH more, whereas oxygen, we know that how the plants give us oxygen, that is through light reactions. Okay, so let us go and revise again. I told you first it starts from photosystem 2. So here in photosystem 2, what happens? Light incidence on the photosystem 2. When the light incidence on the photosystem 2, so the wavelength of light, which is around P680 nanometers of light when it incidents. Now this energy makes the atoms of your chlorophyll or the photosystem 680 get excited. So this excited electrons, what happens? They jump from their atomic orbitals. So when they jump from the atomic orbitals, these energy, okay, when the electrons leave their orbitals, now what happens? These electrons are not used. Instead, they are transported through a series of electron transport complexes. So these electron acceptors or electron transport complexes, what they do? They transport it from P680 to P700. So this happens downhill the redox potential. So once it goes downhill the redox potential, now what happens? It ultimately reach P700. So when it reaches P700, here we have a different type of chlorophyll that that reacts for P700 wavelength of light. Okay. Now we go for the photosystem 1. Here again the light incidence on the photosystem 1. Again the electrons get excited. Okay. So these electrons, what happens? They jump out of their atomic orbitals. Again, these electrons are utilized, okay, are not used, but they're transported downhill the oxidoreductase potential. So once they go down the redox potential, here they ultimately are used for the reduction of NADP plus. So how is NADP plus reduced? NADP plus is added one hydrogen atom to convert into NADPH. So who does this process? NADP NADP, sorry, NADP reductase. Okay, NADP reductase enzyme. Sorry, enzyme. Where is it present? It is present in the stroma. All right, students. So it is present in the stroma. We are talking here about the chlorophyll, right? Chloroplast. Okay, understood? So fine. So now what happens? This reduction happens. Now finally we got the NADPH. All right. So another aspect of our light reaction is how does the ATP synthesis takes place? Okay. How does the ATP synthesis takes place? So we'll talk about it. Now another way in which this photophosphorylation happens is cyclic photophosphorylation. So I know that it might be a little bit confusing. We just talked about non-cyclic photophosphorylation. Now, what is this cyclic photophosphorylation? See, it's very simple. Okay, there are two locations. Okay, one is you have your grana. Correct. We have our grana where the thylakoids are present. Okay. So here in the grana we have both photosystem 1 and then we have photosystem 2. Got it? Now we also know that these grana are interconnected by, okay, a membrane called as stromal lamellae. Stroma lamellae. Correct, no students? There is stroma lamellae, right? So this stroma lamellae, it has only one photosystem. So what photosystem is present in the stroma lamellae? In the stroma lamellae, we have only PS1, not PS2. Understood? So another one that is absent in the stroma lamellae is the NADP reductase. NADP reductase is absent. Very important. Okay, we have to remember. So what are we trying to talk? So there are two types of reaction happening basically by using light. One reaction is where the things go in linear direction. That means there is only one direction to the flow of things. So what is happening here? It has to start at PS2 and then it has to go to PS1. Eventually, this reaction happens. So here, always it has to start at PS2, correct? Whereas when we talk about cyclic photophosphorylation, now the transport of electrons happens through only one photosystem, that is PS1 photosystem, and this type of an arrangement of this cyclic movement of the electrons happens in the stroma lamellae. Okay, reason we have only PS1 and we don't have NADP reductase. Okay, what is happening here? We know that the light incidence on the PS1, and the PS1, once the light incidence on the PS1, now the electrons get excited. Now, here once it gets excited, now there are special electron acceptors like ferredoxin. Okay, so they transport down the gradient. Okay, and finally they transport through ferredoxin, cytochrome B6, cytochrome F, and then plastocyanin, eventually to return the electrons to the same photosystem. Now, as they move the electrons down the gradient, here it is not used for the reduction of NADP. Instead, it is used for the phosphorylation of the ADP molecules. So the ADP is added the phosphate to convert into ATP, which is the energy, which is a currency, right? So this currency of molecules are created by phosphorylation of ADP into ATP. Okay, conversion of ADP into ATP takes place with the help of these electrons, which are moved through these electron acceptors. So where do we get this ATP? We get this ATP when the ferredoxin transports the electron to the next electron acceptor, that is cytochrome B6. There we have one molecule of ATP. And the second molecule we get from cytochrome B6 to cytochrome F. Again, when the electron is transported, we get the another molecule of ATP. Did you understand? So what did we understand here? In our light reactions, we have two photosystems. One is photosystem 2 and photosystem 1, which is participating. So based on how the scheme of transportation of electrons takes place, we can divide these light reactions into cyclic photophosphorylation and non-cyclic photophosphorylation. So in non-cyclic, we understood it is also called as Z scheme. The movement of electrons happen through two photosystems, that is PS2 and PS1, correct? So this movement of electrons happening through PS2 and the PS1 is in a linear direction. That is why we call it as non-cyclic, correct? So what happens in photosystem 2? In non-cyclic photophosphorylation. In non-cyclic photophosphorylation, there is reduction of NADP plus to NADPH with an enzyme called as NADP reductase, which is found in this photosystem. Clear? Whereas in photosystem 1, which is involved in which is involved in cyclic photophosphorylation, only one photosystem is involved, that is PS1. So here the transportation of electrons happen through a cyclic manner where the excited electrons are returned back to the same photosystem. Where is it found? It is found in stroma lamellae alone. Okay. And what does the stroma lamellae lack along with the other photosystem? It lacks NADP reductase enzyme. So that is why the moved electrons are used for conversion of ADP into ATP, okay, through phosphorylation process through an electron transport. Okay, clear? Is it clear? So finally, what do we have? We have NADPH and then we have ATP. And we know what is cyclic and what is non-cyclic. Now, I need to be very sure if you are understanding. Did you understand, students? Yes, no? Come on. JNV Solapur, and then there is Srishant and Pragna, and then we have Priya, right? Are you guys listening? No response? Okay. I'll suppose that you become comfortable. Okay, sooner. Clear? So can we go and see what's the next aspect of our light reactions? Look here. Look here. Very important thing. You have to note down this. You have to note down this. These are the things that you have to remember. What's the difference between cyclic photophosphorylation and non-cyclic photophosphorylation? Cyclic photophosphorylation. What is involved? Only PS1 is involved. Okay. Here, water is not required. We'll talk about it. Water is not required. Oxygen is not evolved. NADPH is not synthesized. Okay, and finally it is used to produce additional ATP, okay, in order to meet the cellular energy demands. Okay, so and the next part of the non-cyclic photophosphorylation is both PS1 and PS2 are involved. Here photolysis of water happens, oxygen is evolved, NADPH is synthesized, and some of the products that are produced in this non-cyclic photophosphorylation are used for the biosynthesis pathway for the production of energy. So anyways, these are the differences, significant differences. Okay, and the next one is we have to understand how the ATP is synthesized. When we talk about the synthesis of ATP molecules, we regard mitochondria as the energy house, okay, of the cell. We call it as the powerhouse of the cell. Correct, right? Why we call mitochondria as the powerhouse of the cell? Because mitochondria provides us with ATP molecules. What are ATP molecules? ATP molecules are energy currencies. For all the cellular activities, we require energy, and the energy demands are met through some of these currencies, we call it as. Okay, these currencies in all the life forms is ATP. Okay, so what is happening here? All the life on Earth is very, very smart. What they do was they understood that if we are able to use any of the components or any of the products which are oxidizable, okay, what did I tell? We if I use any oxidizable component, then I can store energy. How do I oxidize? If I'm able to oxidize something, I'm able to store energy. So nice. It's like your battery, like your phone battery. How you plug it into a charge and the battery charges. Correct, right? Similarly, you're able to store the energy. How? When I oxidize any of the compounds or any of the elements, if I do that, what happens here is the bond formation takes place. So what happens here? The bond formation takes place. So the bond formation is where the energy storage takes place. So how does it work? It's simple. I make sure that I find an oxidizable compound and I will create new bonds. And these bonds, when they are broken, they release energy. And I can use this energy for any of the cellular activities. As simple as that. And our life forms take advantage of this process and they create energy in such a fascinating manner. Okay, so before we understand how did plants end up manufacturing ATP molecules, okay, through a light reaction, okay, we have to understand something more important. So before we talk about the energy, the process itself is depending upon one of the major concepts of utilization of water. So let's see what is happening here. We all understood that the electrons are very, very important factor for the light reactions to take place. So how did we end up with electrons? So we know that the light excited the electrons, and how does the process happen? Here, when the light actually incidents on the photosystem P680 or photosystem 2, the light is utilized where the water molecule is lysed. So we call this reaction as photolysis. Okay, photolysis. So in photolysis, a water molecule is broken down. So when the water molecule is broken down, it releases two hydrogen molecules and half oxygen molecules, okay, along with some electrons. Okay, we know that the hydrogen that is released is positively charged and it is a source of proton. and the electrons that are released by the lysing of the water is used for the transport chain, where NADP plus is reduced as well as ATP is synthesized. These electrons, correct? And the oxygen that is released is not at all useful for the plant, so it just releases, or it just diffuses the oxygen out of the cells, and when it is released into the atmosphere. Now, one of the curious points that you have to understand is, when the plant utilizes the water and it splits the water, it takes up the electrons for one purpose, it takes up the hydrogen as protons for another purpose, and it releases the oxygen. What you have to understand here is, when the hydrogen and the oxygen are split and released, they are not released to the outside surface of the cell, but instead they are released into the lumen. Okay, they're released into the lumen. So if this is the cell, it is released towards the inside. That is the lumen of the thylakoid, it is released. So once it is released into the lumen of the thylakoid, there is some biochemical reaction happening here. Understood? I hope you're following this. In simple manner, I just told you that the light reactions are very, very important for producing two important substances. What is that? One is the ATP, other one is the NADPH. And that all happens with the dependency of water. So the water is lysed or broken down with the use of sunlight and the chlorophyll pigments, where it is broken down into hydrogen, oxygen, and two electrons. So hydrogen becomes a proton source, electrons are transported, and then we have our oxygen that is diffused. Now, the release of these substances happens not towards the outside, but towards the lumen. That means towards the inside of where the reaction is happening, which is something which is quite strange. So now, why does this happen is the real question. If you have understood this part, right, so the next hypothesis, that is chemi-osmotic hypothesis, becomes a cakewalk. And I have no idea how to just move on until you give me some reaction that you have understood. Yeah, students, are you able to follow? Okay. See, actually, this is slowing down our session. So just try to uh, talk. Okay, maybe I will reduce the amount of asking the questions. But what you have to do is just give response to if you're following, if you're able to understand at least the accent, the language, the way of telling things. If not, it's okay. You can just directly tell. We can correct the course because we have very important chapters to discuss in the coming sessions. Okay. So that is why we need a proper channel of communication. So I will leave it to you. And if you have doubts, feel free to ask me. If you don't, it's good enough. Okay. So, so now we understood photolysis and these reactions, correct? Now, how does the energy preparation happens in the light reaction? So that light reaction, as I was talking to you, one of the important aspects. So what did I tell you? I told you that in light reactions, the ATP synthesis takes place, and I also gave you a very decent way of understanding how the life on Earth actually makes energy. So I told you that the making of energy process comes through finding an oxidizable source. So that oxidizable source we have found is the ADP. So we oxidize this ADP into ATP, where the ATP becomes the coin or the currency for energy. So correct, right? So now, how do the synthesis of ATP takes place? So we know that the ATP synthesis is credited to mitochondria a lot. And when we talk about mitochondria, so in mitochondria, we have a system called as electron transport system. Okay, so in this electron transport system, what happens is a proton gradient is created. So we know that when we talk about mitochondria, The mitochondria has outer membrane. Okay, I'll write it here. Okay, it has outer membrane, and then it has inner membrane. Okay, what are we talking about? We're talking about mitochondria, people. Okay, so we have outer membrane and we have inner membrane in the mitochondria. So what do mitochondria do? How does it synthesize energy? It synthesizes energy or ATP by creating something called as proton gradient. Okay, by creating something called as proton gradient. So this proton gradient, that means the difference in the change in the concentration of protons. Okay, so there is a gradient that is created within the two membranes of the mitochondria. Okay, so this proton gradient, which is created within the membranes of the mitochondria, is what helps in the preparation of ATP. We will understand in the process of respiration this how does the proton gradient takes place, if you don't remember or if you don't know. Okay, but our concern is how does the ATP synthesis takes place in thylakoids. Understand this point. So where are the thylakoids are present? The thylakoids are present within the chloroplast. Correct, no people? So where are the where are the thylakoids present? The thylakoids are present within the chloroplast. So now we can divide the chloroplast. Okay, so here we have thylakoids and we have the membrane of thylakoid, right? And we have the lumen. Lumen means that is the inside of the thylakoid. So this is inside of the thylakoid. Okay, this is the membrane of the thylakoid. Okay, membrane of the thylakoid. And I will just erase this or erase. Okay, I will erase this. Let me call it as outside of the thylakoid. Here, this outside space is called as outside of the thylakoid, or I'll call it as stroma. Why do I call it as stroma? If I remember the structure of the chloroplast, correct, right? So I know that the chloroplast has outer membrane, it has inner membrane, and there is grana. Grana is made of coin-like thylakoids, and outside of the thylakoid, this region is called as stroma. And this region, what we are talking is the thylakoid. I hope you are understanding. So now we are understanding how does ATP synthesis take place in the thylakoids. Now I told you that three things, three regions we have to remember or we have to just imagine. One is outside. See, we have, let us say, we have the chloroplast. Within the chloroplast is the uniform stroma. Within the stroma, we can see piles of coin-like structures, which we call it as grana. Correct, right? So grana is made up of coin-like substances called as thylakoids. So if you take the thylakoid, thylakoid has a membrane, and within the membrane, correct, right? Within the membrane, we have inside of the thylakoid, or we call it as lumen of the thylakoid. Okay, we have lumen of the thylakoid, we have outside the thylakoid, that is the stroma of the thylakoid, and we have the membrane of the thylakoid. Now here, the proton gradient is maintained within the lumen of the thylakoid, and we will understand how. Now you have to just remember two important regions. What are those? One is the stroma, other one is the thylakoid. Okay, just wait. Array. So our time is up, students. Can we continue this in the next class because the explanation goes a little bit more? Okay, chalo. I'll take this in the next class. And if you have any doubts, you can ask me now. Okay. So before we talk about the doubts or anything, I will ask you